Hey guys! We're here today to talk about gel electrophoresis. Gel electrophoresis is a method of separating mixtures of DNA, RNA, or proteins according to molecular size, and it's used to analyze DNA sequences. In the labs we'll be doing for the rest of the term, we will use gel electrophoresis to determine the genotypes of individuals. Obviously, we need DNA to analyze a person's genotype, but we only need a specific part of DNA. The DNA placed in gels normally has gone through PCR, polymerase chain reaction. PCR is a process in which numerous copies of a specific sequence of DNA are made. In order to determine the genotype of an individual, we use an enzyme which cuts the DNA at a specific area of the genome called the restriction site. The restriction enzyme functions as a scissor that cuts the alleles in a specific spot. It binds at the DNA and cuts it if it has the enzyme's binding site.
When setting up for gel electrophoresis, make sure that the gates are open to stabilize the gel before placing it in the salt solution. Once the gates are open, place the gel into the salt solution. Make sure the wells are at the black end, the negative end, of the electrophoresis chamber so that the DNA runs to the red. When the electric current is on, the negatively charged DNA molecule will be attracted to the positive end, the red end, of the electrophoresis chamber. After the gel is in place, use your pipette to insert the DNA samples within the wells. The DNA is colorless, but the samples will contain loading dye to help see where the DNA is going when placing it in the wells. After the DNA is in the wells, cover the gel electrophoresis chamber and connect it to the electric circuit. Normally the amount of amps will be given, but they should be around 180 to 120 amp range. Once you run the circuit, you should see bubbles forming on the sides of the chamber, which indicates that the electric circuit is running through the gel chamber.
After we run the gels, you will only see the loading dye in the lanes. So we put the gels under UV light in order for the bands to appear. These bands are the DNA. The CyberSafe in the gel is a UV light reacting liquid that binds the DNA molecularly. When the DNA is placed under UV light, the CyberSafe makes the DNA glow, and you can see the bands clearly. Then we can take a picture of the gel that looks like this.
As you can see, we have different size bands. In the last lane, we have a ladder which we can use to determine the sizes of our DNA and base pairs. A ladder is a set of standard bands with identified base pair sizes that is used to identify the approximate size of a band, and it differs for every gel electrophoresis lab. A base pair is the length of the DNA measured by the number of nucleotides. Shorter DNA fragments with a small number of base pairs will move faster through the gel, and larger DNA fragments move slower through the gel, so the shorter bands will be farther along the gels than the larger ones. As mentioned earlier in this video, the largest band, the band closest to the wells, is the uncut DNA strand, and the cut DNA strands are the two fragments, one being shorter than the other.
It is also important that the DNA fragments are not too small or else they will just run off the end of the gel. Sadly, not every lab can go perfectly. Sometimes errors occur. Common errors include not enough DNA in the sample, primer dimers, error in PCR, problem with the restriction enzyme, or problem with the gel. If you do not have enough DNA in your sample, then you will have a very light band, or you might not have a band at all in the lane. This is because there wasn't a sufficient amount of template DNA extracted from the individual, even with PCR amplifying the DNA sequence. You could also have an error in PCR, which would result in nothing in the lane. Errors can also occur with the restriction enzyme digest. This process has to occur at certain temperatures for a certain amount of time. If this process is not done correctly, then the DNA will remain uncut. Sometimes there is an error with the gel itself. If we suspect this, then we need to look at the ladder to see if it appears any different from how we anticipated. Primer dimers occur when there is not enough DNA in the sample, so primers attach or nail to each other instead of DNA. This will make a hazy band in the gel. Primer dimers tend to be shorter and lower than the other bands in the gel, but not every time. We know this is a lot of information to digest, but we hope that this information is helpful in understanding gel electrophoresis.